I'm Bambos Kyriakou. I'm Professor of Behavioural Genetics at the University of Leicester in the Department of Genetics. We study biological rhythms, which are biorhythms, the 24-hour rhythms that wake us up in the morning and put us to bed at night. And we study them in the fruit fly, Drosophila melanogaster. When I first started working on this was 35 years ago. The medical implications weren't so obvious. But over the decades, it's become quite apparent that rhythms percolate through every aspect of physiology, biochemistry, and metabolism, and behaviour. So everything we do is rhythmic, which is obvious because we live on a rhythmic planet. We have cycles of day and night, and these have been going on relentlessly for 3.5 billion years since life evolved on the planet. So it's not surprising that we've evolved these rhythms. So why is it important? Well, it's important because quarter of our population work shifts. So every week they change their shifts. At weekends they change when they're sociable. They want to be family kind of guys. But on Monday when they start work, they may be working in the middle of the night. And the following week they may advance that by 8 hours, and the week after that they'll advance it by 8 hours. And the trouble with that is that it makes you feel bad. It makes you tired, makes you grumpy. But actually more than that, it actually gets you desynchronized. All your organs have a rhythm, and they've all got to be in synchrony with each other, and if they're not, you feel really bad. It's called jet lag. Some of you will have experienced that. Your brain, when you go to America, resets immediately to local time, but your body doesn't, and that mismatch gives you this sick feeling called jet lag. And shift workers have it all the time, and if you look on any index of their health, they are not healthy people. They have high levels of sleep disorders, depressions, drug addiction, divorce, cancer. And so what we do by studying the clock is basic medical research. The clock is very important because recent studies that we've done in mammals, because we also work on mammals, we found that if you look at all the genes that are expressed in an organ, say the liver, about 15% of those genes are switched on rhythmically. If you look in the heart, 15% of the genes expressed in the heart are turned on rhythmically, but they're a different set of genes. The only genes they have in common are what we call the core clock genes. Everything else is different because the liver has different requirements from the heart or the brain or the intestines. So as you move across organs, you find that almost every gene cycles in a mammal or a fly. So you can see that if you disrupt the cycling of these genes, for example by shift work, it's not surprising that your whole metabolism is not ticking correctly. And if that's not ticking correctly, you start to feel bad and you start to make mistakes. And if you're a jet pilot, that can be really dangerous. Or if you're driving a train at night or a car, that can be very dangerous. It's not just tiredness, it's a desynchrony and cognitive behavior. When you're jet lagged, after a shift work schedule, you'll find that your cognitive behavior has changed. You know, if you do simple tests, psychologists have done this, you see that you don't finish these tests as easily or as efficiently as when you're not jet lagged. So it's very important for every level of functioning, metabolic and cognitive. Well, we use Drosophila A because it's very convenient and it has a generation time of 10 days. In the early 1900s, a guy called Morgan started working on Drosophila, and he and his very brilliant young colleagues worked out all the basic genetics of Drosophila. In fact, Morgan won a Nobel Prize for that work. So we've had 100 years of genetic background in Drosophila. So we know where every gene is on every chromosome. There are only 4 chromosomes, so it's very easy to work with. And the Drosophila genome was published many years ago now, and so it's really easy to work with. We can put genes, we can put genes in to Drosophila. We can take genes out of Drosophila. We can knock down all genes. We can make mutants in every gene that we like, in any tissue that we like. So it's the organism par excellence for doing genetic research, and especially what I do, which is neurogenetic research, which involves looking at the central nervous system, because ultimately that's what mediates rhythmic behavior. It's the nervous system. in the 1980s, people started using Drosophila to clone genes, to get genes in a test tube that were important in making the nervous system work. And the very first behavioural gene that was cloned and identified and isolated in the test tube was a gene called period. And that was about 1984. And period was a gene that I was working on a few years earlier. And period is one of the major genes involved in the clock. When you make mutants of it, you either have a fast clock or a slow clock, or you're an insomniac. And all these mutations are in this gene called period, which happens to be the first behavioural gene in any organism ever identified. And by that I mean that if you remove the gene from the fly, the period gene, the fly's fine, but it's not rhythmic. You put the gene back, it's rhythmic. So it's a behavioural gene, it's not a developmental gene. If you remove it, the fly doesn't die. So it's not an essential gene, it's a behavioural gene. And this was the entree into the whole molecular genetics of the clock, which has now been going on for 25 years, very successfully. And once the genes in the fly had been found, people then looked for those genes in the mouse and the human. And they found them. It took them a while, but they found the same genes in the mouse and the human. And all these clock genes that we first identified in the fly work exactly the same way in the human and the mouse. So the fly is the perfect model for what I do for the human. It's a little human, it's not quite as cuddly, but it's much more convenient to work with. So, I mean, with flies, I mean, you find genes, you identify genes, and usually the way you do it in flies is you make mutants. So you feed flies chemicals, and they damage DNA, that gives you your mutation, and then you study it, and you can study it in behavior or development or on any character you want. But then you want to know whether what you're doing is specific to the fly, or is it more interesting, does it have wider implications? So then you take this piece of DNA and you see whether a similar piece of DNA is found in the human or the mouse. And now you don't have to do any wet molecular work for that. You take your sequence in the fly and you compare it to the human genome, which is published. It was published in 2001. And you will find a gene that's similar to it if it exists in the human. And you'll find it in the mouse as well, the mouse equivalent. The advantage of the mouse is you can knock it out in the mouse and see what happens. So that's really the way you gene hunt. Usually you do it directly by taking a gene in a lower organism and looking for a similar sequence in the higher organism. Sometimes you find genes kind of accidentally in flies, and then you have to identify them in the fly, and that can be quite tricky still, to identify and isolate the gene sequence. But once you've got it, again, you just find it in the human or the mouse genome. And then if you're really interested in it, you get your mice in, you can make directed mutations of that gene in the mouse and see what characters it affects and whether it's the same character you studied in the fly. It's been found, I think, that, I can't remember exactly, but something like 70 or 80% of the genes in Drosophila have a direct human equivalent, and most of those have a disease associated with their mutation. So the fly has become a wonderful model for just about every disease going. For example, in my laboratory, we also study Huntington's disease, which, as many of you know, is a neurodegenerative disorder. And you can make Huntington's disease in flies. And if you make Huntington's disease in flies, you can then make mutations in other genes to see if they can actually make the Huntington's flies better. And that's another big project we have in the lab. I do that with a colleague of mine here called Flavio Giorgini. It's a kind of an interesting project because the way we do it is first we make Huntington's in yeast. And Huntington's you can make in yeast and the Huntington cells die. But you can rescue that if you make another mutation randomly in the genome. So sometimes you can rescue these dead Huntington's yeasts with this other mutation. So then you think, well, how does that apply to humans, for example? Well, we can't jump straight to humans, but what we can do is make Huntington flies and then test those suppressors of Huntington's that we get from yeast in the fly. And what we've been finding is that many of these suppressors of yeast Huntington's suppress fly Huntington's. In fact, one of these is now undergoing drug trials in mice. So that has a direct application to medical research. You know, we found a gene We know what drugs might target this gene, and these drugs are now going through clinical trials in America with our collaborators. So that's, you know, pretty direct evidence for how useful flies can be. One of the most striking studies on humans was a family where they had this strange character segregating in different generations. And these people, they weren't really ill. They just woke up really early and they went to bed really early. So they would be in bed by 6 p.m, whereas the normal population is in bed by 10 or 11. And they wake up really early. And this was clearly genetic because it went through every generation of this family. So it looked like it was what we call a dominant gene. And these people were just, they had different sleeping habits. And if you put them in isolation, so they had no cues, no light, dark cues, their clocks ticked fast with about a 22-hour rhythm. Very interesting. So pedigree analysis was done, and that includes molecular analysis. And it turned out there was a single mutation in one gene which was causing their clock to run fast and give them this early bird phenotype. They were larks. So they got up early, they went to bed early. Guess what the gene was? It was human period. So it was the same gene that was first identified in the fly. So the first clock gene in the fly was period. The first clock gene in humans was period. But it's more interesting than that. In the fly, the mutation affected a single amino acid, a serine. It's one of the amino acids, in a particular position. The mutation in the humans affected the same serine. So it was in the same position, this mutation, in the fly and in the human. And that was really very, very exciting. So again, just underscores the fact that humans are just like flies. It was very interesting that a few years ago during the American election, Sarah Palin, a somewhat glamorous woman who was going to become president, she hoped, or vice president, I can't remember, was it vice president? She made a speech where I think she was talking about autism in children and spending money on autism research, which obviously is very important. Then in the next sentence, she said something like, people are actually working on fruit flies now. Can you believe that? What a waste of money that is. I think the actual words were, I kid you not. And the funny thing was that autism researchers actually benefited from fly research. And there's one gene in fruit flies that is actually believed to be a candidate gene in autism. But a lot of the general public and politicians, if they want to score cheap points, will look at money spent on research like in fruit flies or yeast or other model organisms and think it's a waste of time. Actually, it isn't, because, as I've said before, the fly is a wonderful model for medical research. And it's being used for all sorts of things, like immunology now. It's a very good model for immunogenetics. It's a model for every kind of disease you can think of that affects humans, or every kind of behavior. It's a great model, and it's cheap to work with. So the money that's spent on research in fruit flies saves a lot of money, because if you were to do the same research on mice, and granted some research doesn't pan out, so I mean, if we knew what we were doing, we wouldn't call it research, would we? Okay, so a lot of research projects don't work because they're a risk, they're a gamble. But it's much better to spend the money on Drosophila and find it hasn't worked than spending a lot more money on mice to find that it doesn't work. So you're actually saving money.